Okay, so moving on to one of our general types of tissue, epithelial tissue. I'm going to break this lecture up into two different recordings because it's a little bit longer. So first we're going to talk about what is epithelial tissue. So this is sheets of cells that are going to cover our body surfaces or line our internal cavities. So basically anywhere that's exposed to the external environment, whether it's inside of us, like through some sort of canal or tubing, or the surface of our body, like our skin, is going to be composed of epithelial tissue. There's two main forms. So we have the covering and the lining epithelial tissue that I just referred to. And we also have glandular epithelia. So this is the tissue that lines all the glands in our body. So for example, our salivary glands typically form boundaries whose main functions are going to be protection, absorption, filtration, excretion, and sensory reception. As we look at the different types of specific epithelial tissue, they encompass some of these functions and not necessarily all of them. You do not need to know what the specific functions are for the specific tissue types. Just have a general idea of the functions for all epithelial tissue as a whole. So when we're talking about epithelial tissue, they have 5 distinguishing characteristics. They have something called polarity, which we'll talk about, specialized contacts, where they're making between epithelial cells. They're always going to be supported by connective tissue. So underneath epithelial tissue layer, directly below it is always going to be connective tissue before we have any other tissue type. They're avascular, so they don't have any blood supply at all, but they are innervated, meaning that they do have nerves. And they're able to regenerate themselves. So let's first talk about polarity. So all of the cells that make up the epithelium, so epithelial cells, have polarity, meaning that they have 2 surfaces. So, and both these surfaces are going to differ in their structure and their function. So the first surface is the apical surface, and this is the one that's exposed to basically an empty space or to the lumen or the inside of some sort of cavity, to the tubing. So they're always going to see like a white area whenever you're looking for epithelial tissue, because it's always going to have this apical surface that's exposed to some sort of opening. Along the apical surfaces, they can have some specialized features. So for example, on some of the columnar cells, you can see microvilli, and those are just these finger-like projections that are going to increase the surface area. Most apical surfaces are smooth though. If we're talking about the basal surface, this is the opposite side. So the basal surface is connected to the basal lamina, and we'll see in a little bit how this is just basically a small separation between the epithelial tissue and connective tissue. Because remember, connective tissue is always going to be underneath epithelial tissue. So the basal surface is going to be the one that faces inwards on the body, and it's going to be attached to a basal lamina, which is eventually going to help connect it to the underlying connective tissue. So here's a picture. So right here we can see our epithelial layer. It looks like it is simple columnar. The apical surface is going to be the one that's bordering all of this open space out here. And the basal surface is going to be on top of the connective tissue. So always connective tissue underlining it. In this case, it looks like it's areolar connective tissue. We can see a little bit, we'll talk about in a second, the basement membrane that's between the epithelial tissue and the connective tissue, but you can't really see it under a microscope. You can only really distinguish between the 2 general tissue types. You can also see, because this is columnar, very, very faintly, the microvilli out here. So it looks like little tiny fuzziness at the end of at the apical surface of these simple columnar cells. So the second characteristic is specialized contacts. So because these cells are adjacent to each other and form kind of like a continuous sheet, they have some contact with each other. When we look at connective tissue, they don't have connections or contacts with each other. They're kind of just dispersed throughout the connective tissue itself. So they're not singular rows or singular sheets. They're just sporadically found within the tissue. So epithelial tissue, again, because they're connected, they have these specialized contact points that helps bind all of the epithelial cells together. So where they make these contacts, they have two specific types of junctions that we've talked about before. They're going to have tight junctions. Remember, that's kind of like your Ziploc analogy, and that's really just to prevent things from linking between the two cells. They also have desmosomes. Your analogy for that was like Velcro, kind of sticks together. It's going to help resist tension so that the cells are not pulled apart. So if you think about the epithelia that makes up your skin itself, we don't want those to be pulled apart, so we need those desmosomes. We also kind of want to waterproof ourselves, so we don't, if we're in a pool or swimming or doing anything, that we don't absorb all of that water. So we want those tight junctions as well, so we don't allow things to cross our barrier that we've formed. The third characteristic is, like I mentioned, they're always supported by connective tissue. So underneath the basal layer will always be a layer of connective tissue, one of the different types. But we already talked about, we had the basal lamina, which is directly underneath the basal surface. So here's our apical surface of our epithelial tissue, our basal surface. And this is all of our epithelial tissue. And then we have this little thing that we call the basement membrane, and it's made up of the basal lamina, which lies just deep to the basal layer. Jeez Louise. And I don't know why that keeps doing that. Okay. And then we have a reticular layer underneath that. Collectively, the basal lamina and the reticular lamina make up the basement membrane. And then directly below the basement membrane that's separating the epithelial tissue, we'll have some sort of connective tissue. And the whole purpose of the basement membrane is really just to help reinforce the epithelial sheet and help resist stretching and tearing between the two tissues. So it helps connect them together. We talked about another characteristic being avascular but innervated. So avascular means that there is no blood vessels found in the epithelial tissue. However, if you think about tissue itself, anytime we're replicating or dividing our cells, we need some source of nutrients, and we get the nutrients from blood. So they still need a supply of nutrients and a blood supply, but they're just getting it from the underlying connective tissues, because the connective tissues underneath do have blood vessels. So then they can release the nutrients that can diffuse up to the epithelial tissue. So even though they don't have any blood vessels, they do have nerve fibers. So if you pinch yourself on the arm right now, you can feel it. Another thing to help you remember about it being avascular, if you're thinking about your skin being epithelial tissue, if you cut yourself, and we'll talk more about this in the integumentary system, like say a small paper cut, sometimes you don't bleed. And the reason why you don't bleed when you get that paper cut is because we don't have blood supply in that stratified squamous epithelia that we have. If we cut and we do pretty deep, say we accidentally nick ourselves with a knife and we go deeper, then we hit the underlying connective tissue, which does have a blood supply. So if you cut yourself and you don't see any blood, you've just damaged or cut through your epithelial tissue. If you do see blood, that means you've gone all the way through your epithelial tissue and down into your connective tissue where the blood supply is. The last characteristic is regeneration. So they are able to regenerate themselves. Again, if you think about our skin cells are constantly being sloughed off throughout the day based off of our activities. So they have to be able to regenerate in order to reproduce our linings or protective mechanism. So that's going to require adequate nutrition and cell division. Again, they're going to get their nutrients for cell division from the blood supply from their underlying connective tissue. Another thing when we talk about epithelial tissue, it's always a first and last name for how we name it. So the first name is going to indicate how many layers thick is this epithelial tissue. If it's a single layer thick, we just say it's simple. If it's multiple layers, meaning 2 or more, then we refer to it as being stratified. The second name or the last name of epithelial tissue is going to name the shape of the cell. And more easier to look at or to name cells looking at the nucleus itself and the shape of the nucleus rather than the outline of the plasma membrane of each of the cells. So second name indicates the cell shape. So squamous is going to be really, really flat. It looks like a fried egg, if you think about that. Cuboidal is going to be cube shaped, but the nucleus is very spherical. And then columnar, these are going to be really elongated or tall column-like cells, and their nuclei is going to be oval. So we're looking at, I don't know why it keeps doing that. So we're looking at like an oval nucleus like that. For columnar, we're looking at a spherical, pretty symmetrical, round nuclei there. And then for squamous, we're looking like really squished, kind of like that. However, if we have stratified epithelia, I'm going to show you on the picture here. Stratified epithelia is always made up of 2 different cell shapes. So if we look at this one, we see like we have some cuboidal down here. And then if we look at the apical surface, we see that we have squished nuclei, so squamous cells. So how do we know how to name it? Well, you always look at the apical surface, and that names the stratified epithelium. So if we look here, because I see these squished nuclei along here, I'm going to say that this tissue is stratified squamous. Doesn't matter what's underneath it, you're looking at the outermost surface or the apical layer to name it. So in general, when we talk about single layered epithelia or simple epithelia, those are going to be involved in absorption, secretion, and filtration. And that makes sense because we're talking about a single layer of cells. So we will allow things to basically absorb, be secreted, or filtered through this membrane. If we had a stratified epithelia, it'd be really hard to absorb substances across it or secrete substances or to filter substances because it'd be hard for those substances to actually cross multiple layers. So simple epithelia, these are their functions because they're only a single layer thick. So if we're talking specifically about simple squamous epithelia, this is predominantly found in your lungs. You can also find some in your kidneys, but I'm worried about one particular organ where you can locate this. And in this case, for your lecture exam, is going to be the lungs. We do have 2 special simple squamous epithelia, and we name them accordingly. So we have endothelium. This is the lining of lymphatic vessels, blood vessels, and the heart. So when you hit AMP2 and you hit the cardiovascular system, they'll talk a lot about the endothelial cells that make up these vessels. Then you have the mesothelium, and this is going to be the epithelia that makes up the serous membranes in the ventral body cavity. So I would kind of be aware of the 2 specialized types of simple squamous epithelia. Here's an image. This is a great image. This one looks exactly like what the lung slide looks like. So again, here you're worried about lungs is where this can be found for the lecture exam. And we can see it's a single row. We don't see a thick layer. And we also see these kind of squished or flattened nuclei. The next one, simple cuboidal. Simple cuboidal typically lines ducts. So they make up the walls of small ducts. And many kidney tubules. So when you talk about the urinary system later on, and you actually have to find these tubules, you're always going to look for simple cuboidal going around in a ring. They're typically involved in secretion and absorption. Again, I'm just worried that you understand the function of epithelial tissue as a whole. And they're going to be single layer cells because it's simple. So this is what simple cuboidal looks like. The big place here is your kidney because we have so many tubules, small, tiny tubules that make up the nephrons, which make up the kidney. This is kind of what the cross-section of 90% of the kidney section will look like. So we can see these rings right here. Each one of these is a tubule. Within the kidney, we can see right here, this white space in the middle is what we call the lumen. So that's where the actual tube is. And then the wall of the tube that they're talking about for the kidneys is made up of simple cuboidal. So we can see we have these pretty spherical looking nuclei right there, and they're in a single row. So it's going to be simple cuboidal. The next one is simple columnar, and again, these are just going to be a single layer of tall, and their cells, they're going to be pretty closely packed, as with all epithelial tissue. They do have two modifications in some of the cells. So some of the cells can have microvilli, so that's one of the modifications. Others can have cilia. The big difference is if we're talking about microvilli, those are going to be to increase the surface area, and cilia are really just, they function to move substances in one direction across the tissue surface. Some of the tissues will also contain mucus secreting goblet cells. So I'll point one out if we see one on one of these pictures. Again, because we're talking about simple, epithelia is going to be involved in absorption and secretion. Additionally, because we have these goblet cells, it's going to be involved in secretion specifically of mucus, enzymes, and some other substances. So the big place where you're going to want to understand where simple columnar is found at is the digestive tract. Our digestive tract has huge amounts of simple columnar because we have to be able to secrete this mucus and enzymes in order to help break down all the substances as it travels through our alimentary canal. So we can still see right here this little fuzzy area. This fuzzy area is called the microvilli. We refer to it also as the brush border because it kind of looks like a broom or a brush at its tips. We can tell it's simple columnar because we see a single row going across here, a very oval-like nuclei. Here we can see a couple goblet cells. So here's a goblet cell, and I can't tell if that's one. Those two are goblet cells for sure that are going to secrete that mucus. The next one's going to be pseudostratified columnar epithelium. Pseudo meaning it gives a false appearance of being stratified, but in reality it's actually simple, so it's a single layer. What gives it its pseudostratified look is that the cells are actually varying in height. So it looks like we have these stacked nuclei and these multiple layers, but in reality we don't. The nuclei are in various locations because not all of the cells have their apical surface reaching the same height. A lot of the cells are going to be ciliated, and because we're dealing with columnar, we're also going to be involved with secretion, mucus, because we can have goblet cells in this area. So where are we going to find this? Our trachea is a huge place, especially when we're talking about the ciliated version, because we have to be able to secrete mucus in order to, again, propel stuff through our respiratory system. And we also want the mucus and the goblet cells in our trachea so that we can trap any type of particles that we're inhaling during inspiration during that time. And here's another example of a goblet cell, so they're not always perfectly spherical. We can see that this cell right here is one cell, and we have one nucleus. Over here we have another one that's going to reach that height in a nucleus, and because they're not all reaching to this surface out here, their nuclei appear in various locations, which gives them that pseudostratified look. So now we can move on to the stratified epithelia. So this is the most common and widespread type of epithelia that we have, especially when we're talking about stratified squamous. So our skin is stratified squamous. Free surface is always going to be squamous. Underneath, a lot of times you'll either see cuboidal or columnar. But again, because we're naming it based off of the cell shape at the apical surface, no matter what other types of cells we see underneath, we're going to say it's stratified squamous. It's going to be in areas where there's high wear and tear. So our skin is going to be a main one. We can also have keratinized and non-keratinized stratified squamous epithelia. So if we're talking about keratinized, that can be on our skin, and non-keratinized would be in our internal cavity, so something like our vagina. The keratination is really just a hard, tough pigment, so we would expect to have a harder pigment in areas exposed to the external surface, like our skin. So here's an example of non-keratinized stratified squamous. This is going to be like in our esophagus or our mouth or our vagina. We do have keratinized if we're talking about our actual skin. And we can see up here we have squished or flattened nuclei. Down here it looks like we might have some cuboidal and even some columnar. So we know we have multiple layers, so it has to be stratified. We look at the apical surface and we see it's squamous, so we say it's stratified squamous epithelia. Then we have stratified cuboidal. Both stratified cuboidal and stratified columnar are very, very rare. So your textbook didn't include any images. I found these and pulled these from the internet because I wanted you to have an understanding of what they look like, because we do have some examples in lab on the histology exam. So they're typically found in sweat or mammary glands, not always. They're kind of sporadically interspersed within the tissue. And when we say stratified cuboidal, it's not multiple layers like our stratified squamous. Typically, it's only 2 cell layers thick. How you're trying to identify the stratified cuboidal is looking for stacked nuclei, meaning we have nuclei above and below each other. So if we look here, I can see that we have stratified cuboidal because I look, the nucleus is pretty round. We have one there, we have one below it too. So we have nuclei above and below each other, which indicates that we have stratified epithelia. Stratified columnar, again, very, very rare. We have it in some places, very, very small amounts in our pharynx as well as our urethra. So if we look here, we can tell it's columnar by looking at the nuclei. I can tell it's stratified because I see nuclei here, here, and here. So we have multiple layers of nuclei above and below each other, which indicates that it's multiple layers, not just a single layer. Also, if you look, we can see that each of the columnar cells reaches to the apical surface, and we have a nice even row. We don't see any that are ending down at the basal side. So we say that this is truly stratified columnar epithelium, not the pseudo-stratified columnar. The last one's going to be transitional epithelium, and this is typically found in areas where we have a lot of stretch. So for example, your bladder. Your bladder is the main one here. The easiest way to tell this one is it looks like that you have a mixture, again, of columnar, possibly some cuboidal, columnar, cuboidal. But if you look at the apical surface here, instead of having these elongated cells like this at the basal side, you look at the apical surface and now we see horizontal cells. So these horizontal cells and the elongated cells down here, the columnar ones, that's going to help you indicate that this is a transitional epithelium. And again, the big one here is going to be your bladder because they're found in areas and organs that need a lot of stretch.